Hey, it's October 5th, and this is the second video with the SORS-B grant that Ben and I have been working on. And so far, we haven't done any of the testing yet, but Jason and Steven have gone over the techniques and a lot of the processes and instruments that we're going to be using in order to study the samples and data better. So the first process we've been looking at is PCR, or polymerase chain reaction. The premise of PCR is to be able to produce large quantities and amplify a target DNA. The point of this is to be able to do more testing and to look more closely at a specific region rather than a whole DNA strand. The first part in PCR is to denature the DNA strand. This is usually done at around 95 degrees Celsius. After this, the DNA is annealed, meaning cooled down and broken into separate strands. Then primers are attached onto each one of the two separate strands at a specific location wanting to be targeted. After this, polymerase is added and uses nitrogen base pairs floating around in order to fill in and replicate the targeted region. Finally, this process is completed in many cycles until there is enough of the targeted DNA. This is called exponential amplification. Another process that we have learned about is TACMAN, which deals with copy number variations. In TACMAN, there's a target assay, which is the region that you are studying, and a reference assay. These two assays are eventually added into a tube along with the master mix. Then 4 samples are poured into a sample plate, and then this is transferred into a PCR. Both of these processes help to look at specific parts of the DNA and see, if any, the effect on autism. By using two different methods to look at similar information, it ensures that the data is reliable and not because of the effects of the process. It is important that the data received is accurate. Accuracy is achieved by using many different processes, many different tests, and making sure all procedures are followed.